Hi everybody, welcome to our talk about database search identification algorithms for protein and peptide identification. This is one of my favorite topics, and it's one that I've invested a lot of papers and time in. So if I run away from you, if I'm moving too quickly, I want you to grab an ankle and slow me down, because I want to make sure everybody understands this. Database search is the fundamental method that we use for identifying peptides and proteins. So without further ado, I want to go ahead to split up our discussion today to three topics that I feel like we really have to cover. The first of these is how we would compare MS scans to peptide sequences drawn from a sequence database. This is the fundamental operation of the database search algorithm, and understanding how that works is fundamental to everything that happens later on down the line. After that, we're going to spend a little bit of time talking about how we figure out which matches between peptide sequences and tandem mass spectra we actually trust. And for that, we'll be talking about false discovery rate and some other methods that exist for deciding which IDs we're going to keep and which ones we're going to throw out. And then for the third part, we're going to talk about the inference of proteins, or the assembly of peptides to produce protein sequences. I think that this will be a little discouraging for people who are very new to the field, because often we trust the proteins that come back from database search without really thinking about how we got to them. So I'm hoping that we can talk about the concept of parsimony and how that relates to the proteins that we infer for a data set. I want to start with a little bit of analytical chemistry. There's a kind of a broad misconception in our field that the process of CID somehow explodes peptides, that it makes them blow up, that it breaks, that it's so energetic it just smashes apart a peptide bond to create fragment ions. And this is kind of a misconception that I think makes people ignore the chemistry of what's going on. So let's start with this, with defining CID. Collision-induced dissociation is abbreviated the way it is because it's to remind us of the physical process that allows us to break peptides. Collision-induced dissociation is causing the peptide to collide with gas molecules. These gas molecule collisions are happening rapidly enough that the peptide ion is heating up in the vacuum of the mass spec. So what does it mean to be hot in a vacuum, right? In the case of peptide ions, it means that they've got more energy than they normally would. And that energy causes the peptide to begin vibrating in unusual ways. So remember that we're adding protons to these peptides before we put them into the mass spec. I mean, we've got them in acid, basically, and we've put them through a very strong positive charge in the electrospray source. And that means that we've got a proton sitting on board these peptides. Okay, so that proton usually sits in a nice, cozy, basic site. Normally that's a nice arginine side chain. I mean, that thing looks like a big catcher's mitt if you look at it from a chemical perspective. Or a nice lysine side chain, which is convenient given that we're cutting at arginine and lysines. But there's also the possibility that you may have a proton riding along on the N-terminus of the peptide, because the N-terminus of a peptide is a primary amine. So those protons start moving around when we energize the peptides in this way. And occasionally, you'll get one that sits down on a carbonyl. Now, I'm sure you all remember and love organic chemistry. And so you remember that a carbonyl is a C double bonded to an O. So imagine a proton sitting down on that O, in that spot where we've got that little red one there. Now, you got to remember that those double lines represent electrons holding that oxygen to that carbon. You get a little bit of cheating going on. When you add a nice positive charge to that oxygen, the electrons start scooting up away from that carbon, and that leaves the carbon a little bit positive, just a little bit. That creates an opportunity, because if you remember your organic chemistry, you probably remember having a couple dots to represent an electron pair that's kind of sitting around not doing much, and you can get an attack from one carbonyl to the next that creates this very briefly lived intermediate, a ring structure within the peptide that dissociates to create a B ion and a Y ion. I want to make one other point about the B ions here, that this Y ion over at the right looks very much like a small peptide, right? We've got our NCC, NCC, all very good. But the B ion doesn't look like a small peptide anymore, does it? That's the detritus, basically, of this circularizing reaction. So you end up with what's called an oxazolone ion. And B ions usually take on this oxazolone form. Now, there are some unusual things that oxazolone ions can do, and especially as we move to things like higher energy collision-induced dissociation, the HCD methods, or beam-type fragmentation, what you have in triple TOF, you'll see that these B ions can go on to break in more interesting ways. A B4 can become a B3, a B3 can become a B2. B2 ions have their very own chemistry, so they can become very, very stable. So I want you to remember that what's happening to produce these fragment ions is a chemical process, and which amino acids you have where is going to have some implications for how they break. Now, in the slide, I say that most of the fragment ions result from the breakage of peptide bonds. That is not necessarily the only way that this can happen. You may have a B ion that throws off carbon monoxide to form an A ion. So the A ion is not formed directly by snapping a peptide bond. It's a B ion that's ejecting carbon monoxide through another intermediate. It is also possible that the peptide ion itself can undergo some disposal. Maybe it throws off water, or it throws off ammonia, and that can create a neutral loss ion. Generally speaking, though, we think of these tandem mass spectra as consisting of a bunch of B and Y ions. So you can see that this peptide has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 amino acids. And we see that I've drawn in a breakage after the fourth. So this creates an N-terminal ion containing the first 4 amino acids, which we call a B4, and we have the last 6 amino acids coming off as a Y6. The number just refers to how many amino acids are in that structure. Okay, so we can notice in this case that this particular breakage would create a B ion that has the mass of 415.2, and a Y ion that has a mass of 572.36. Now, does everyone know precisely what math we use to get at the computed M over Z value for one of these B ions or Y ions? Okay, well, then it's a great place to start. Everyone, I think, has started memorizing that amino acid residue table, right? Great, I'm going to start with an easy one. How about this I? I stands for what amino acid? Isoleucine, great. All right, and isoleucine has what mass? 113, did I hear? You didn't say 113. Well, that's okay. I'm going to tell you it's 113. And that's really important because it's a mass it shares with leucine. And leucine and isoleucine are ridiculously common amino acids, so 113 is a good one to know. What about serine? Anyone know that one? Okay, well, there's a little memorization activity to do, I can tell. So serine is 87. Now, threonine is just 14 more than serine, so 87 plus 14 is 101. Give that man a cookie. Pass those down. All right. You can add together the amino acid masses, and that will get you close to the B ion or Y ion mass. But you need to remember a couple more things. In the case of a B ion, you need to add a number of protons representing its current charge. So if you're predicting the singly charged B4 mass for this, you add together the residue mass of T, S, I, and add 1 for the single proton charging them. It's a little different on the Y side of this. Here we need to add together those 6 amino acids that are included in its sequence. We would need to add 1 because we're predicting its singly charged form, but we also need to add what? No, not a bad guess. But the primary amine is gaining a hydrogen, and the C-terminus of it is adding a what? A hydroxide. So if you add the hydrogen you're adding at the N-terminus to the OH that you're grabbing from the C-terminus together, that's water. So a Y ion is the sum of the amino acids plus water, 18, plus the number of ionizing protons. Okay, so this tells us how to say, does a fragment ion exist in this tandem mass spectrum? But we also have series of fragment ions. So you can imagine if this breakage were to occur 1 amino acid to the left towards the N-terminus, instead of having B4Y6, you would have B3Y7. Great. Okay, which means that the Y7 ion should differ in mass from the Y6 ion by the mass of isoleucine, which we just said was 113. Okay, so 572 plus 113. Someone do the math for me. 572 plus 113 is 685. 685. All right, so we see that the Y6 ion, matching to this 572, is matched. It would lead on to this next amino acid, the next ion in the series, from Y6 to Y7 of 685. Okay, so this spectrum has evidence for multiple breakages throughout the peptide sequence. The B4-Y6 pair is what we're pointing to, but there are others in the spectrum that back up the other peptide bond breakages. Our ability to trust that we've got the correct peptide sequence matched to the spectrum depends on how many of these peptide bonds have evidence in the fragments of that tandem mass spectrum. Okay, normally I do more talk about analytical chemistry up at the top, but I really want to spend time on algorithms today since we talk about analytical chemistry frequently in this group. So let's talk about what you did on the bench to produce your data, because we're going to reverse that as we deal with the bioinformatics from this collection. We started with a mixture of proteins. We used trypsin, most typically, to digest it. That produced maybe 10 times as many peptides as we started with proteins in the first place. We did some separation, like reverse phase chromatography, liquid chromatography, collecting data-dependent tandem mass spectra from it, and now we have our tandem mass spectra. So having done all these steps, we now need to reverse them on the bioinformatically to make sense of what we've got. The first step, coming across the bottom here, is to identify each tandem mass spectrum to a set of peptide sequences that are most likely to explain the appearance of that spectrum. So essentially, you can think of each spectrum coming back with a list of a result from the database search engine that says, I think this sequence is the best explanation for that spectrum. But we don't trust them all. We don't routinely identify more than, say, 50% of all the spectra we collect. So we need to have a kind of a moment of doubt. We need to say, well, the database search engine was just doing the best job it could with this spectrum. But is the evidence that it found strong enough for me to accept that peptide identification? That's the process that gets us from a pile of PSMs, peptide-spectrum matches, to a set of peptide sequences that we really trust. And then from that, we're going to infer a set of proteins to explain the peptides we've seen. All right? So we started on the bench with a bunch of proteins, digested the peptides, produced tandem mass spectra, and now we're going to produce a bunch of PSMs, figure out which peptides we actually trust from that collection, and infer the proteins back. This diagram is probably the critical one to get in order to understand database search. Because these pieces are kind of complex and important, we're going to go into several of these individually. But I want you to remember that, as an early paper in the field said, we're looking up the answer in the back of the book. We are not, in general, interpreting tandem mass spectra based on the spectrum itself. Instead, we're starting with a very large set of hypotheses. And those hypotheses are to say, this peptide sequence is represented by this tandem mass spectrum. But we have, in some cases, tens of thousands of possible hypotheses to explain a given tandem mass spectrum. Those folks who are working with low-resolution mass specs, like quadruple ion traps, it's routinely the case that tens of thousands of possible peptide sequences could be compared to an individual spectrum. So we need to have some way to judge that. This diagram shows us how to get there. So we're going to start with a FASTA sequence database. This is just a big text file, but it's a listing of all the different proteins that are known to be possible for this organism. Now, did we get those from proteomics? We did not. Most generally, we got these from annotating the genomes of the species in question. We haven't yet identified all of the peptides and all the proteins in humans, right, from proteomics experiments. So it's a case where we've learned what protein sequences exist for humans by annotating the genome. So we might start with that set of protein sequences as our input. If the protein of interest is not in the lookup table that we've created, if it's not in the sequence database, we cannot identify it by database search. All right, from here, in silico, in the computer, we need to digest up this protein sequence to see what list of peptide sequences could result from that protein. So in silico, we need to be able to say what peptides would result if we use trypsin on this protein. Next, in database search, we need to specify in advance which modifications are possible for us to identify. So very frequently, we use what's called a static modification in the case of iodoacetamide. Does everyone remember what iodoacetamide does to our proteins when we digest them up? It's carbamidamethylation. Yes, carbamidamethylation. So we've used something like dithiothreatol to break apart disulfide bonds, and now we want to block those sulfides from reforming those bridges. So we use iodoacetamide to block them off, and it creates what's called a carbamidamethylation. So normally, the mass we use for cysteine is 103 Daltons. But we tell the database search engine, forget about its original mass. We're not going to use the mass of 103 for cysteine. We're going to assume that all cysteines have been blocked in this way. We always tell it to use 57 mass units higher, or 160 for the mass of cysteine. That's a static modification. But a PTM, the post-translational modification, typically means that we're allowing for multiple possibilities for an individual amino acid. In the case of phosphorylation, we would probably say serine, threonine, and tyrosine. Any one of those three amino acids must be considered in two forms, unmodified and phosphorylated, plus 80, in other words. Okay, this leads to a big expansion to a set of decorated peptides. It is not the case, however, that every one of those peptide sequences must be compared to every tandem mass spectrum. Everyone get that? How would we decide which peptide sequences should be compared to tandem mass spectra? Precursor mass, exactly. It's right there in the diagram. So we didn't measure just fragment ions for these peptides. We also measured precursor masses. So typically we have a high resolution MS scan that shows us to a very, very small degree of error what the mass to charge value of the ion is. And if we saw multiple isotopes for it, well then we can probably infer its charge, thus making it possible to just infer its mass directly. Okay, so the precursor mass filter says that we're only going to compare to each spectrum peptide sequences that are very, very close to what we observed for this peptide sequence. And especially when you have high resolution MS, the number of candidates that you need to compare to every spectrum falls precipitously by having a very accurate measure of its mass. Okay, now we have a peptide sequence with modifications perhaps, and a tandem mass spectrum. How do we lay one of those against the other? That's kind of a hard thing to do. So there are two routes you can go with this. One is that you can infer sequence from the tandem mass spectrum and see if that inferred sequence matches the sequence database. That's an approach called sequence tagging, and we're not discussing it today. We're instead talking about the thing that we do all the time, which is to predict what the spectrum should look like if it were this peptide sequence. So for a given peptide sequence, we can predict a set of B ions and Y ions, and then we need to have some way in the end to score what we observed in the tandem mass spectrum versus what we predicted from the peptide sequence. Any questions so far? Now we're going to come back to several of these steps because I think it's really important everyone gets them. Let's start with this problem of emulating the behavior of a protease in silico. Up at the top, I've drawn a protein sequence, and I've drawn some gaps in it, as you can see. Those gaps represent arginine and lysine residues. So trypsin should cut every time there's an arginine and a lysine, but not really. There are some exceptions to this, right? So, Brandon, when would you anticipate that trypsin might fail to cut at an arginine lysine? Proline. Okay, right. So proline has some weird behavior in these structures, and it can prevent the cutting of an arginine or lysine that's right next door to it. That's one. Are there others? Sure, modification. Yes, if you have an acetylation stuck on a lysine, it's quite unlikely that lysine is still going to be accessible for digestion. You may have a run of arginine and lysine residues, like RK or something like that. You're not going to cut each and every one of those. So there are lots of things that can cause trypsin to behave a little weirdly. So we've tried to take those into account when we emulate in silico how these things work. But let's start with the easy case. It may be that trypsin has cut at a arginine on one end and a lysine on the other end, and now we have a fully tryptic peptide. That's great. There's another case for that shown up here as well. See this missed cleavage peptide? It ends at a legitimate cutting site, and it starts at a legitimate cutting site, but it also has an uncut arginine or lysine inside it. So missed cleavages can also happen, and they happen with some frequency, in fact, so worth knowing about those. There's also a large number of peptides called semi-tryptic peptides, especially when you have samples that are dominated by just a few proteins, or samples that have a lot of endogenous proteases doing stuff. Seeing semi-tryptic peptides can be a bit of a catch-up, so I want to make sure you're aware that they do in fact happen sometimes. So it may be that it's a peptide that starts at a legitimate trypsin cutting site, but that doesn't extend all the way to the next one. Or it could be that it starts somewhere in the middle here and runs to a legitimate trypsin cutting site. So semi-tryptic peptides are only identifiable if you tell the algorithm, look for them. If you don't tell it to look for them, you won't see them. And you might mistake those spectra for something else as a result. Finally, there are frequently some very large regions of proteins that we fail to identify peptides from. Imagine a transmembrane domain of a protein. What is the nature of amino acids used in transmembrane domains? Anyone know? What's their dominant characteristic, their chemical phenomenon called hydrophobicity? Hydrophobicity. If you have a protein that's passing through a membrane, it's passing through a very hydrophobic region. So typically, you're going to see a whole lot of amino acids that have hydrophobic side chains. A transmembrane domain is very unlikely to have a whole bunch of arginines and lysines packed in there because those are very polar and very basic amino acids, so you're just not going to find them. If you have a region like that, you may have a fairly long stretch of amino acids that simply doesn't get digested. Okay. Now, I mentioned that we need to consider the possibility that a residue, an amino acid, may be present in unmodified and modified form if we're looking for PTMs. We can think of this as the one that we always include, right? Oxidation of methionine. We look for oxidation of methionine because methionine, you manipulate your protein in the tube on the bench. You look at it wrong and suddenly it's oxidized, right? So we need to consider the possibility that methionine may be in its normal form or it might be in an oxidized form. However, let's return to this question of phosphopeptides. Here I've grabbed a short peptide sequence out of alpha-casein from cows. So we note that we have 3 different sites within this peptide sequence that match our phosphorylation definition. This serine, that serine, or the threonine over here, any of those may be modified or may not be. Okay, so when we look at the expansion that takes place for this peptide, with just the possibility that serine or threonine or tyrosine could be phosphorylated, we see that one peptide has expanded into 8. We have the completely unmodified form at top. We have 3 possibilities representing single phosphorylation. We have 3 possibilities representing double phosphorylation, and 1 possibility representing triple phosphorylation. So you can think about this as a math problem. Each of these peptides has each of these amino acids has 2 possible states, and there are 3 of them. 2 raised to the 3rd power is 8. So that's how one peptide became 8 in this case. This is why it's expensive in time and expensive in sensitivity as well to search for lots and lots of PTMs. because one peptide can explode into this very large number of peptides, creating an exponential search cost. Okay. Now, did I see a question? Okay, I thought I would check. All right. Next, let's get on to peptide mass filter. I have to say that you guys who have come into the proteomics field in a time of high-resolution mass measurement have it so much better than the very bad old days. In the days of the first instruments sold explicitly for proteomics, things like the old LCQ and stuff like that, I mean, yeah, the triple quads could also be used for it back in the day, but when people really started going hardcore for proteomics, they'd start using LCQs for this. And the LCQ could produce 2,000 tandem mass spectra in the space of like an hour-long gradient. And we were like, that's amazing! It was so much better. The other thing about them, though, was that their measurements were all imprecise mass accuracy. So you would say that it had unit mass accuracy. In a tandem mass spectrum, you could trust if it said that the peak was located at 788, that its true value was anywhere as low as, I said 788? It could be as low as 787.5 or up to 788.5. And we thought that was great. Things are so much better with high resolution. But it was much, much worse when we got to the precursor mass accuracy. Because you see, we had this problem that the instruments had pretty low resolution, and frequently your measurement of an individual peptide ion was dealing with a whole bunch of other ions all kind of crammed in to give us this weird convolved signal. Imagine the case of a +2 peptide at low resolution. The peptide monoisotope and the peptide monoisotope were too close together, the monoisotope and the C13 peak were too close together to be resolved by that instrument. And so you end up with this massive blob of intensity, and you kind of had to guess what the precursor M/Z was. So down here at the bottom, I've put kind of the implications of that. And these numbers are arguable. This is back of the envelope stuff. In the old days, when we were searching with LCQ or LTQ data, we would typically allow for a mass error of 1.25 M/Z for precursors. You just had to do this, or you were going to exclude the correct answer from your comparison list. That's rough. I've written time of flight. Mass accuracy, we typically operate with something like 50 parts per million. That's probably far too skeptical for today's instruments. Frequently, you'll find that the type of mass correction and mass calibration is taking place in these instruments so that mass-corrected values are being written in the file. In a case like that, 50 ppm would be too wide. I think you could get by with, what, 20 or 30 on most triple tops? 20 or 30 ppm these days? They're very nice instruments. Now, frequently, we're dealing with mass accuracy that's come out of a Fourier transform instrument or an Orbitrap style instrument. And in cases like that, you have so much more mass accuracy to work with that typically you don't have to consider a peptide if it falls further than 10 parts per million from the measured M/Z of the peptide. That's brilliant. So I've tried to represent that by this blue bar. If this blue bar is nice and wide, it means you have to consider more candidates as you try to interpret this tandem mass spectrum. If it's nice and narrow, because you've got a nice Fourier transform instrument producing that mass, well then you're in much better shape. Now this red bar is kind of a different case. It is often more likely that the instrument will make an error in picking the correct monoisotope than that it will give you a more than 10 part per million measurement on that. So being off by 1 Dalton is much more likely than being off by 0.1 Dalton. Frustrating, but real. So it is possible that you may need to tell the search engine that you're willing to allow for off by 1 or even off by 2 errors in allowing it to slip 2 neutrons over for choosing the monoisotope. In a case like that, you are now considering, you're multiplying the number of candidates you're comparing the spectrum to by the number of different isotopes you're willing to consider. But it's a common option now in a lot of search engines. I want to stop for just a moment to talk about how we model what a spectrum should look like for a given precursor. This is not something we talk about all that frequently when we're talking about search engines, but it's a really important issue. When we generate the list of fragment ions we expect for a particular peptide sequence, we have a much more complex prediction in the case of a +3 or higher peptide than we do in the case of a +1 or a +2. In the case of a +1 or a +2 peptide ion, when it fragments, we anticipate that that one proton will go with each side, or in the case of a +1, that either it goes this way or it goes that way. I mean, in either case, you get a +1, a single +1 produced by that breakage. So when if you were to predict a 10 amino acid peptide in say +2 form, you would have how many different peptide bonds you could break? It's a trick question. 10 amino acids, how many peptide bonds connect them? 9, and you said 8. Do you have a reason why you said 8?
I looked at the picture in my brain and my brain was confused.
Ah, okay, well there is a reason. I already mentioned that, that you get the most common types of breakages of peptides when the prior carbonyl attacks across the one in front of it. There is no carbonyl to attack after the first amino acid. So you have 10 amino acids, 9 peptide bonds. The normal reactions imply only 8 of those would actually be broken. Okay, so we can just start then with this assumption that for a 10 amino acid peptide, 8 possible breakage sites, and we can predict then a B ion and a Y ion for each of those breakages. So that means a 10-mer leads to 16 fragment ions we anticipate, 2 ions for each breakpoint. All good? I don't want to just run away, so okay, good. So we have basically twice as many fragment ions observed as we have peptide bonds to break. That is different, however, when we get to the +3 scenario. What possibility did we ignore as we broke apart a +2? So yeah, we ignored the first peptide bond. What was yours? Where the charge was going to go. Very good. So I used to do a talk about peptide fragmentation, and I had a slide up there called the divorce. For anyone who's ever been a close witness to divorce, you know that there's always a battle about the stuff. And in the same way, when a peptide bond breaks, there's this brief moment where these two fragment ions are going at it, hammer and tongs, deciding which one is going to abscond with the charge. So it's possible that as a doubly charged peptide breaks, that one proton goes with each side. But it is also possible, especially when you have a very lopsided break near the end of a peptide, that one fragment will take both charges with it. You can see that database search engines generally ignore that possibility. They say, if we don't identify singly charged fragments from a doubly charged peptide, we don't care. Now, in a +3 case, this situation of the divorce gets even worse, because now we have 3 peptides riding on board a peptide, 3 protons riding on board a peptide, and as it breaks, one of them is most likely to get 2 charges, and the other one is likely to get 1 charge. It is possible to get a +3 fragment from a +3 precursor, though. So very weird things can happen. So in general, database search engines will predict each B ion in 2 forms, in singly charged form and doubly charged form, and every Y ion in singly charged form and doubly charged form. I'm going to check my time, make sure I'm running way over already. Okay, so how we predict fragment ions is generally very well conserved among all the different engines that we use. How do you decide in the end that a predicted spectrum looks like what the observed spectrum looks like? So we have, at this point, 2 spectra. It's just one was generated in silico. We predicted what this sequence should look like if it were a tandem mass spectrum. Sequest cross-correlation is one of the oldest scoring methods out there, and there are lots of search engines out there that make use of this method. You do not have to be using something that has Sequest anywhere in its name, and you could still be using a cross-correlation scoring system. I want you to imagine that one hand represents the spectrum we've predicted from a given peptide sequence. On the other hand, literally, we have an observed tandem mass spectrum, and we want to decide if our model for what the sequence should look like represents what we saw in the observed spectrum. So I want you to imagine I can run one of these, slide it past the other like that. You see that? Now, there's a point that we call 0 offset, right? Where the value I observed at 1,000 matches for the values I modeled at 1,000, right? That's tau 0, or no offset. But we also have these these negative tau positions and these positive tau positions. And we can evaluate how the alignment looks between the model and the observed spectrum at all these different positions. So here at the bottom right, we have this figure from the original Sequest paper, 1994, that shows how the alignment looks as you rub one spectrum past the other for various values of tau. Now you'll note that there's a spike right here at 0. That spike, how much it stands out above the surrounding hairball, is an X-core score. The offset score at 0 gets compared to the alignment scores nearby, and basically I forget if it's the ratio or the difference between the two, is what gets reported as the score in the X-core of Sequest. Now, I would note that many years after this 1994 paper, Jimmy Eng realized that there was a simplification to the math in computing X-core that made computing it about 10 times as computationally efficient. That was published in 2008, and that model is still used in things like Comet, for example, and Sequest HT. So for all those years, in those 14 years, imagine how much faster people could have been finding their results for their data sets if only that math error had been spotted sooner. Awkward. I'm aware that a lot of people here are familiar with the MaxQuant algorithm from Matthias Mann's lab. And I wanted to talk about Andromeda because it's an important example of a peak counter approach, a match counting approach. Now, did you see anything at all in the Sequest math that reflected that we cared about how many peaks we matched between the two? We didn't. We just cared how much better the signal aligned as we got to this tau 0 value. But Andromeda follows a model that's really common out there for evaluating the quality of the match, and it's based on how many hits are produced between the spectrum and the model. So you can instead of thinking of producing a model spectrum for what this peptide sequence should look like, you can think of it as an M/Z line with points marked out. I think a peak should appear here. I think a peak should appear here, one here, and one here, and one here. Right? So our model has become one-dimensional. Now we ask, at these locations where we think a peak should appear, did we or did we not see it? And having predicted some number of ions, you remember we had a 10-mer and it produced 16 fragments that we predicted. So we predicted these 16 locations, we count how many of them were actually occupied by peaks, and we can use this math to compute a score for that comparison. So I'm going to let us imagine that we went looking then for 16 fragments and we observed 10. The way that this score works is to compute the probability of randomly matching 10 of those peaks. Then it computes the probability of randomly matching 11, randomly matching 12, randomly matching 13, 14, 15, 16. Those probabilities added together are a p-value saying how likely was it that we would match this many or more peaks for this particular model. Okay, and they use an approach called the binomial model for computing that. There's a lot more that's going on as well, though. The software takes into account that there's the possibility of neutral loss. What if, for example, your Y ions also had water loss ions in them? What if you had a phosphorylation and you saw the loss of the beta elimination of that phosphorylation? These are examples of neutral losses. The software optimizes that score, saying which one gives us the less probable random match, with neutral losses or without, and it chooses the one that drives the score higher to a lower p-value in essence. It also tries to set a sliding scale on how many peaks is it going to consider real peaks. You know, you might have measured 200 peaks in this particular tandem mass spectrum, but they have a distribution, right? Some of them are really high screamers, you know there's a peak there, and others are just down there in the grass. So the software is going to use a sliding scale, trying to decide what it's going to count as a peak in order to optimize that scoring match. So both of these optimizations are going on to see what maximizes its score. There are strengths and weaknesses to this model. It's not my favorite. But then if you really want to get to the wild and zany world, you can look at something like the MSGF plus search algorithm. MSGF plus is quite unusual in that it is not matching against the spectrum as observed. Instead, it's trying a partial interpretation of that spectrum model. Instead of looking at a peak as a given M over Z value, it is trying to put together complementary pairs within the spectrum and neutral loss information within the spectrum in order to put together what we call a prefix residue mass graph. I've drawn that into the slide here. So effectively, a PRM graph is a different representation of the spectral information to say, this peak represents the probability of a break this many mass units from the N-terminus. It's a rather weird thing to think about. So if you have a big spike at 57, that's its argument that the spectral data argue for an initial glycine in the sequence. So it's doing this partial inference from the tandem mass spectra, produce PRM graphs, and its matching takes place in the context of the PRM graphs. And the score that comes out of it is called an expectation value, to say that if we did this search at random, out of all peptide sequences, what score would we have expected to happen by random chance, right? So this is a really unusual search engine, but they've shown quite a lot of power in coming up with correct answers for tandem mass spectra. It's particularly valuable if you have low resolution measurements of fragment ions.
So what is the optimal e-value?
Well, 0, obviously. When you have a e-value rated metric in bioinformatics, you're always hoping for lower is better. To say what number of results would I have expected by random chance to have produced a score this good, 0 is your best case. I mean, driving the limit is 0. So when you run a blast search, if you get an e-value of 1 from your blast search, that means that by random chance you would have expected the best result to be exactly this good. You want that to be nice and low to suggest that under no circumstances would a random search have produced a hit this good. Yeah, good question. Okay, database search scores, however, have 2 different roles. We've talked about how we push the correct sequence to rank number 1 for a tandem mass spectrum. And that's of course very valuable. We want to be able to show that for a given spectrum, we can score the real sequence better than anything else in a database. Generally speaking, if we don't get if the correct match doesn't make it to the top for a particular spectrum, it's lost. That spectrum's information is just lost. We don't really spend a lot of time looking after second-ranked matches at present time. There are some systems that do, but generally speaking, if only the top sequence is considered further by most downstream pipelines. All right, so let's talk about that second role, though. If you have well-calibrated scores, you can use these scores not just to decide which sequence is the best hit for a particular spectrum, but also to help you decide which spectra do you trust the matches for. Now, at this point, I'm going to start using the phrase PSMs a lot. And it's really important that everybody understands what they're good for. A peptide-spectrum match refers to a hypothesis about the identity of a given peptide. We have a tandem mass spectrum, we have a peptide sequence that's been associated with it by the database search engine, and we now have a hypothesis for what this spectrum means. It is this peptide sequence. So this PSM has come about through database search. Now we need to figure out which PSMs we trust out of a large set of spectra. Okay. So we need to be able to figure out which spectra are very worthwhile for us to work with and which ones are not. There are 2 main approaches that have come out for making sense of these. One is distribution modeling, and the other one is this Target-Decoy or reverse search approach. Both of these are very popular. So we would start by saying that distribution modeling like peptide profit is really, really good when you need to have very good resolution of which PSMs you trust and which ones you don't. So we're going to put a score on an individual PSM to say, this is how much confidence I have in this particular match. Target-Decoy is a much more aggregate approach to say that for this set of identifications, what level of error do we expect? What proportion of error do we expect? Question? Okay, all right, we'll move on. One of the things that people who do distribution modeling care about a lot is the combination of lots of sources of information. In the original paper on peptide profit, way back from 2002, we see that the cross-correlation score from Sequest was taken into account. But it also takes into account the delta CN score. Now, if you haven't used Sequest a lot, this score might be a little unusual for you. So I'd say that the cross-correlation score should be, under a given set of configurations, the same every time you compare this peptide with this spectrum. A delta CN score, on the other hand, says how much better was our top hit than the second best hit. The notion being here that if you're well ahead of number 2, you have more confidence in an ID, whereas if it's just eking out a little bit over number 2, that's not as compelling. We had a preliminary score rank. This was a kind of a pre-score that gets computed by Sequest. They found that it was useful to include that in the final scoring. And then we have this DM term, and this one is actually really, really important for many, many people who are using the peptide profit route. By the way, keep the cookie bag moving. I know people over here are hungry. You're not hungry? Well, I'm hungry. Eat one for me. All right, well, okay. He wants a cookie. That's good. All right, so the delta mass function is really, really valuable. In the case of accurate mass measurement of peptides in particular, things like for a transform or TOF instruments, you can trust a peptide sequence that's very, very close to what was observed more than you can trust one that's way out at the limit of what the precursor mass filter would allow for. And so this delta mass term, DM, ends up becoming quite important in how we discriminate these things. All of these scores get combined together to create one discriminant score, which you see on the x-axis here, that's used in peptide profit. So you can imagine then that all of the PSMs are along this distribution, and this overall curve, the sum of these two distributions, is the set of all the discriminant scores. Now, we see that the negatives all kind of lump together here at relatively low scores. But we also have this hump to the right of what are called positives here. These are the correct PSMs. So that's pretty great. It's lovely when the data look like this. But I would point out that way back in the day, in 2002, identifying 10% of your spectra was considered pretty good success. Today, we would scream and cry if that happened to us. So yeah, in a modern HCD set, you should expect that the number of positively identified spectra is going to be a lot larger than what you have in the negatives. In any case, peptide profit believes that this is two distributions of scores overlaid on each other, summed together, in fact. The negatives have this lower mean, and the positives have a higher mean. And sometimes they have different distributions as well. Frequently, the negative distribution is kind of bounded by this there's no worse score than this, and so they slam up against the wall. So you might use two different kinds of distributions to model the two humps here. In this case, the positive was modeled with I actually used to have it in here, a normal distribution, while the negative was modeled with a gamma distribution. So the software needs to find a way to fit these two curves to this empirical score distribution that it's got in order to maximize their fit. From that, you can now say for a given PSM, I have a score of 2. What is the probability that it comes from the positive distribution versus the probability of all of them, basically? So we have two models fit to it, and we can compare the probabilities under both of those distributions to evaluate its probability of correctness. Now that's quite a lot more complex than the simple Target-Decoy method that a lot of people use. And complicated can mean good and it can mean bad. And there are times when you're going to want to use something like peptide profit, and there are times when you are not. So in a lot of the cases, we use a Target-Decoy approach. In Target-Decoy approach, we make the whole database search worse in order to be able to characterize error at the end. The rationale behind this is that if you have an incorrectly identified spectrum, it has an equal probability of matching to a sequence from the forward database or from the reversed database. So we can ask how many matches we had to known incorrect sequences and say to ourselves, but for every one that we know matched to an incorrect sequence, there was also a false identification to a forward sequence. So by sorting all the IDs from top to bottom, we can figure out where to set a threshold in order to compute that we've hit a false discovery rate for the whole collection above that threshold that we actually want. So maybe we say a 2% PSM false discovery rate would be acceptable. We'll move that bar downward until we've hit that 2% boundary for the whole collection. We get to the last, oh yes sir.
Just on the, or not the use, but some search engines, they require like a decoy set
Yes.
as part of the FASTA.
Yes.
And I'm just thinking of MSFrag in particular, that or rather FragMap that uses peptide profit after MSFragger.
Yes.
That requires a decoy set. But peptide profit uses this discriminant score. So how do they fit together? What's the software doing?
Yes, so you can imagine that peptide profit could simply assert that this distribution is two distributions summed together and try to fit them to it. But it's a whole lot easier if it's able to pin down where this negative distribution is based on the information in the search. So there was an update to the peptide profit published, I think 2007, if I'm not mistaken, where the algorithm was modified to allow decoys to be used to position this negative model very accurately, because you know that every one of the decoy hits is false. So you can use that to position this distribution very sharply and even evaluate whether the distribution you've chosen matches the score distribution that you actually see empirically. So yes, it is possible to use the decoy information even in the context of a peptide profit model. Good question. All right, now that I've given myself only 5 minutes to finish here, so I'm going to start by saying, if you're really interested in whole protein identification, understanding which PTMs are appearing in concert with each other, you really need to be doing top-down proteomics. Because the stage of moving from a bunch of peptide identifications to protein identifications is one of inference. If you have a phosphorylation on this peptide over here and a phosphorylation on that peptide over there, and they happen to match to the same accession protein in the database, you still have no guarantee that these two modifications exist on the same protein structure. So you hear what I'm saying with that? All right, so these peptides don't remember which protein they came from. They've been separated from each other through trypsin. They've been separated from each other through the column, the RPLC. The tandem mass spectrum has no information to give you about which protein or which proteoform this particular peptide comes from, which means that everything past there becomes inferential. So how is it that it's not obvious which protein a particular peptide comes from? There are three most common reasons that pop out for this: orthology, paralogy, and isoforms. And to be very brief, you and I are related to blades of grass, which is, I think, a very nice way to think on a weekday afternoon. We are all but blades of grass. The proteins that you see in grass for dealing with sugars are very much like the proteins that you have in your bodies for dealing with sugars. This is an example of orthology, that you see the same gene in multiple species performing the same function. If you deal with a multi-species database like UniProt, which is a very frequent approach, it's quite likely that a given peptide could match to proteins from all kinds of species because things like actin are strongly conserved across species. So this is an example where orthology could lead to the same peptide matching to any number of protein sequences. Paralogy is a separate kind of problem, because over the evolutionary history of species, we have lots of cases, especially in things like trees, trees are murder, where gene duplication has been taking place in a lot of cases. Sometimes we find that over evolutionary history, a gene that originally coded for an enzyme continues to code for that enzyme, but now a duplicate of it has independently mutated away from it and takes on an altogether different kind of role in the body. So it may be, in even something like the human genome, you're going to have cases where the same peptide appears in many paralogous copies that occurred through gene duplication over our evolutionary history. Finally, lots of protein databases include different isoforms representing different transcripts from the same gene. In a case like that, each transcript is going to have a lot of shared content for the parts of the gene structure that all the transcripts go through. But as soon as you get to exons that differentiate the transcripts, you have different peptide sequences. So for that, you will find that these conserved regions across the transcripts from a given gene will be hit multiple times because each transcript gets listed separately in the database. Now I'm going to show you a really ugly case where all of these actually came into play. So in this case, we are looking at a xenograft. We have grafted a human cancer, we've injected human tumor cells into a mouse, and that mouse has now grown human tumor material in its abdomen that's being fed by mouse vasculature. So when we attempt to identify proteins from the xenograft, we have to take into consideration the possibility that we have either mouse proteins or human proteins grown together in one tissue. That's really problematic here. So I'm showing what's called a bipartite graph. Bipartite meaning basically two parts to it. At the left, we have different protein hypotheses. At the right, we have different groups of peptides. And these edges drawn between them reflect cases where a given protein could be used to explain a given peptide. So you can see that these 10 peptides in the second blob at the right could be explained by the second protein, or they could be explained by the third protein. Either of those would be perfectly good. We also have cases where many proteins all have exactly the same peptides in evidence. Look at the set down here at the very bottom in blue. Table 1Y human has 3 different protein sequences in this database, all of which match to each of these 4 groups of peptides at the right. You need to have some sort of guide for how to disentangle this. In the bad old days of the 1990s, you could get away with claiming that all of those proteins were there. But today, reviewer 1, 2, and 3 will all mess you up for it. So, how do we protect you from that? We need a concept of parsimony. We need to have some way of grouping identical proteins to say these proteins, any one of them, would be an equally good explanation for why we saw these peptides. That's a case of indistinguishable proteins. We also need to be careful about things like subset proteins. If you have a protein that matches peptides A, B, and C, and another protein that only matches B and C but not A, it's best to strike out that second protein because its evidence is a subset of the evidence for the first. These are examples of the kinds of rules that we need in order to be parsimonious with our data. A parsimonious explanation is one that invokes fewer proteins because they have equal explanatory power. So to use this example, you will see that only 2 of the 7 protein groups at the left have double boxes around them. If you follow all of those edges, you can see that including just these 2 double-boxed proteins gives you the ability to explain all of the peptides at right. One of the consequences in this case is that what looked like evidence for both human and mouse proteins suddenly becomes explained only by human proteins. The mouse evidence just falls right out. And I want to note here, we're not in the business of proving negatives. We have not proved by this that there were no mouse proteins present. We just showed that these 2 human proteins were a sufficient explanation and a minimal explanation for what we saw. Parsimony rules in general can have a very big effect. Many people have used the, here called the Sigma 49, but the Sigma Universal Proteomics Standard, now with 48 human proteins. So we shot this into our instrument, we searched it with all of SwissProt. And SwissProt is a bad database in a lot of ways, right? Hugely informative, very valuable. But if you just search for all of SwissProt, you're really setting yourself up for trouble because of the amount of orthology you're going to encounter. So every time we saw peptides that were shared among different species, we jacked up our protein count. Here, we started with 49 proteins in the mix, and we detected more than 400 proteins. Laughably bad. This is not what happens, right? So we need to be careful about grouping together indiscernibles and applying parsimony in order to get our true protein counts down to where they belong. We knew that we shot 49 proteins into the instrument. When we use grouping plus parsimony, we see that the numbers that come out correspond correctly with what we shot into the instrument. So proteomic identification depends upon a database of known protein sequences. If you're working in a species where you don't have a set of known protein sequences, you are in need of a non-model organism specialist. Feel free to chat with me in your spare time. We need a set of known protein sequences in order to pull out our lists of peptides that will get compared to each tandem mass spectrum. Controlling error in the set of identified PSMs is critical to reliable proteomics interpretation. If you have filtered your data so loosely that you have a 10% protein FDR, you're probably in a world of hurt, and you're going to have lots of false discoveries there to trip you up. Detecting indiscernible proteins and applying parsimony is a conservative practice, but it prevents you from going beyond what your data can tentatively support. Applying parsimony rules is necessary to having a good inventory. Okay, so those are the three parts: database search, FDR control, and protein inference. We all rely on them every day, so it's really, really important that you understand them, and that will protect you as you try to understand what your data mean. I'm glad to talk with any of you about this at any point. This is obviously a subject I care about a lot, so feel free to come ask me anytime I'm over here. This video will be on YouTube, so I hope that it is useful to further generations of grad students. Thank you.